Exploring SNP in human, the missense variation RS1801133 in the human MTHFR gene has been linked to elevated levels of homocysteine, an amino acid whose plasma concentration seems to be associated with the risk of cardiovascular diseases, neural tube defects, and loss of cognitive function. This SNP is also referred to as A22V, alanine 222 valine, as well as other HGVS names. Part A of the exercise has asked us to find the page with information for RS1801133, our variant of interest. To find information about this variant, we're going to search for RS1801133 in the search box, and we're going to click Go. We're going to choose the first search results, which is the human variant. We're now on the variant tab. The variant tab shows us the most severe consequences of this variant, the associated alleles, location, co-located variants, and some more information. Part B of the exercise has asked us, is RS1801133 a missense variation in all transcripts of the MTHFR gene? What is the amino acid change? To find the answer for exercise B, we're going to click on Genes and Regulation on the left-hand side. The page now shows genes and transcript consequences table. Since the exercise has asked for the missense consequence, we're going to filter our table by the missense consequence type by typing in missense. Our filtered table shows us that RS1801133 is a missense variant in 9 MTHFR transcripts. It appears twice for each transcript because of two alternative alleles A and C. If we scroll back to the table, we can see that for the A allele, the amino acid change is A to V, and for the C allele, the amino acid change is from A to G. Part C of the exercise has asked us, why are the alleles for this variation in ensemble given as G/A and not as C/T as is in the literature? The answer to this exercise is that in ensemble, the alleles of RS1801133 variant are given as G/A, which is shown here, because these are the alleles in the forward strand of the genome. In literature, the alleles are given as C/T because the MTHFR gene is located on the reverse strand. Part D of the exercise has asked us, what is the major allele of RS1801133? To answer this part of the exercise, we're going to click on Population Genetics on the left-hand side. We're going to scroll down to Population Genetics and focus on 1000 Genomes Project Phase 3 allele frequencies. The bar charts show us that in all populations but one, which is the Colombian population, the allele G is the major allele, whereas in the Colombian population, G is represented in 45% of the population and A is represented in 54% of the population. Part E of the exercise has asked us, in which paper is the association between RS1801133 and homocysteine levels described? To find this information, we're going to click on Phenotype Data on the left-hand side. The specific studies where the association was originally described is given in the Phenotype Data table. Links between RS1801133 and homocysteine levels are described in 3 papers that are mentioned here. We can click on these PubMed IDs for more details. Part F. According to the data imported from dbSNP, The ancestral allele for RS1801133 is G. Ancestral alleles in dbSNP are based on a comparison between human and chimp. Does the sequence at this position in other primates confirm that the ancestral allele is indeed G? To answer part F, we're going to click on Phylogenetic Context on the left-hand side, and we're now going to select an alignment. From Alignment Selector, we're going to click on Multiple Alignments, and we're going to select 12 primates EPO. Once we've selected our multiple alignment, the selected species are going to appear on the right-hand side. Once we're happy with our selection, we can click Apply. Our alignments have now loaded on the page, and we can see a region containing the SNP, highlighted in red and placed in the center, and we also have its flanking sequence displayed. As we can see here that all other primates also have a G at this position, confirming that G is the ancestral allele